Okay, time to break down some carbs and get some ATP out of it. So, the definition for glucose catabolism isn't on this slide. Hold on. It's part of your study guide. I know exactly. That's why I'm moved ahead. Okay, so most of our carbs we break down using for energy. So, for glucose catabolism, we are oxidizing or breaking down glucose into carbon dioxide. So, the whole, if you want to look at the equation for it, taking glucose and oxygen and turning it into carbon dioxide and water. So that's why we breathe out carbon dioxide. So that's just the words of that same equation. So the function of this reaction is to transfer the energy from glucose into ATP, and then this is just the excess waste that we create in that process. Here's your glucose catabolism definition. So these, and this is the same. This is just in kind of paragraph form. This is in bullet form. So glucose catabolism is a series of small steps, each controlled by a separate enzyme in which energy is released in small amounts, and then that's transferred to ATP. The rest is released as heat. So 3 major pathways of breaking down glucose or glucose catabolism. So the first one is glycolysis. So remember on that first slide where you have these big chunky arrows? So that first arrow where you're going down toward our process of making ATP, that one is your glycolysis. So there's a whole processing system that you got to do before you can even start really getting into the actual process of making ATP, which is glycolysis. So we're going to take a glucose molecule, which has 6 carbons, and break it down into 2 pyruvate molecules, which have 3 carbons. Now, in this whole process, we only make 2 ATP molecules. But we can then use those pyruvate molecules either in anaerobic fermentation or aerobic respiration. So depending on your oxygen levels, determine on which method you use. So for anaerobic fermentation, this is when you have a lack of oxygen. The pyruvate is reduced to lactate, and this allows for a repeat of glycolysis where you can make 2 more ATPs. However, the more efficient model is aerobic respiration, and that is done in the mitochondria when you have plenty of oxygen. So pyruvate is oxidized instead from pyruvate to your carbon dioxide and water, and in that process you make 32 ATP. So lot more efficient. Here with our anaerobic fermentation, once you start getting enough oxygen in, you can actually go back. Your liver can convert the lactate back to pyruvate, and then you can switch to aerobic respiration to get your full amount of energy that you can suck out of a glucose. Okay, so during this process, enzymes are going to remove off electrons in the form of hydrogen atoms and then transfer them to coenzymes, and they keep passing them along. They're passing along this chain of different other enzymes in order to set up the production for making ATP. There are 2 different coenzymes that help with this pass along, and that's NAD+ and FAD. So just showing you because you're going to see them again. So when our hydrogen atoms are removed off of which starts off as our pyruvate molecule, they're removed in pairs. So 2 hydrogen atoms which have a proton and an electron. So these pieces are transferred to a coenzyme. And then they keep getting passed along until they're able to set up a gradient for making ATP. So our coenzymes, FAD and NAD+, can actually bind to these pieces in order to transfer them. So we're going to actually look at this whole process of these different players. So again, our glycolysis, that big chunky arrow that's going downward from glucose. You don't have to know all these steps, but we're going to cover some of them. So in our glycolysis, our cell is going to convert our 1 glucose molecule into 2 pyruvate molecules to net 2 ATP molecules at the end. Then we take these 2 pyruvate molecules, and depending on your oxygen levels, either go through anaerobic fermentation and a repeat of glycolysis with low oxygen, or go through aerobic respiration in the mitochondria using the citric acid cycle. And again, 2 ATP versus 32. So for glycolysis, so we are lysis or breaking down our, and technically it's glucose, but don't ask me why it's glycolysis. glycolysis for this, but anyway. So, glycolysis, we're taking our glucose and eventually going to the process of going to pyruvate. So, there's multiple steps. I'm not going to cover them because I don't talk about them on the test. But one of them I'm going to mention is phosphorylation. That actually helps you to allow to bring more glucose into the cell, because now this molecule is changed just enough where you're not building up a concentration of glucose in the cell. So that allows you to continue to bring glucose into the cell. Okay, doke. So again, more different steps that I'm going to skip because I don't care. Eventually, your 6-carbon glucose molecule becomes pyruvate. So, during this process, 2 net ATP molecules are created. Some are still attached to our coenzyme. So, in this case, it's our NAD+, which now has hydrogen attached to it. And then we also have these 2 hydrogen protons that are just kind of hanging out by themselves. And then we're still left with our 2 pyruvate molecules. So we still got a lot of energy stored in all these different molecules and have barely made any kind of ATP. Okay, so depending on how much oxygen you have determines the fate of our pyruvate. So again, absence of oxygen or mitochondria, such as in your red blood cells, then you have to generate ATP using glycolysis, so using anaerobic fermentation to regenerate your NAD+ coenzyme so that you can repeat glycolysis and make 2 more ATP molecules from that. So, when you go through anaerobic fermentation, the pyruvate is oxidized into lactate, reduced to lactate. So that lactate still holds a lot of that energy. So that lactate can then go to the liver. The liver oxidizes it back to pyruvate so that when, again, you get enough oxygen, you can finally go through aerobic respiration and actually really extract out energy from that molecule. Another function that the liver can do is to take that lactate and create it into glucose 6-phosphate, so convert it back, and then be able to use that glucose 6-phosphate for glycogen for storage and then release the glucose when necessary. So I'm actually going to go back to that slide again. So here we have our pyruvate, but instead of taking that pyruvate through, so pyruvate going into our anaerobic fermentation, but then that can lactate can then be converted back to pyruvate, and we can go this backwards function, which we'll get to this later, the gluconeogenesis, back to our glucose 6-phosphate, which then can be stored as glycogen. So being able to use different pathways in order to essentially get what your body needs and maximize the energy that you have. So not being wasteful of our energy molecules. Okay, so drawbacks to anaerobic fermentation. So again, you're going to be making lactate from the pyruvate in this process. Unfortunately, lactate is toxic. Again, this is why it has to go through the liver to be converted in either back to pyruvate or to something else that our body can use. So in our muscles, or particularly our skeletal muscle, it is very tolerant of anaerobic fermentation. So as you're exercising, you do have some stores in your skeletal muscle to make ATP, but eventually they're going to get used up. And so you switch to anaerobic fermentation. That's okay for that type of muscle, but not for your cardiac muscle. So that lactate, that's partially what's causing your pain signals. Because again, lactate is toxic. So if your heart has to switch to anaerobic fermentation because there's not enough oxygen because you cut off a blood supply because you're eating such fatty foods, so this blocked artery not getting enough oxygen, having to switch to anaerobic fermentation, and therefore that's the pain signal during a heart attack. The brain, however, is not able to switch across. It just won't make the ATP, and that's why lack of oxygen to the brain just passes out as an I quit, and when you give me oxygen, I'll come back online. Okay, so most of our ATP is created in the mitochondria. Again, you have to have oxygen The reason why oxygen is your final electron acceptor. So we're actually going to go into that section. So in our aerobic respiration, our pyruvate is then oxidized for our aerobic respiration. So further breaking down the pyruvate to really extract out the energy. So you have two different sections of reactions. We have our matrix reactions. So that's on this inner portion. So our mitochondria has two layers. We have an outer layer and an inner layer. So in the very middle of our inner layer, that's our matrix. So there's some reactions there. And then there are some reactions that occur on the inner membrane across it. And that is, we set up a gradient and then we use that gradient to power our enzyme to make ATP. Okay, so using our chart of our pathway. So here's our pyruvate, blah, 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 don't care. We get down to a 2-carbon molecule called an acetyl group. That acetyl group is going to bind to coenzyme A to form acetyl-CoA. That acetyl-CoA is going to drop off 2 of those carbons in attaching it to oxaloacetic acid. It's just a 4-carbon molecule. You don't have to know that name. So this is going to go back to a 6-carbon molecule, which is citric acid, which is your first molecule for the citric acid cycle. If you're older, this used to be called the Krebs cycle, if you just happen to remember. Yeah. Okay. Yeah, there goes that light bulb. Okay, so in this whole process, again, blah, 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 I don't care. We're actually going to, you don't have to know all these little independent, little tiny little things there. But some of these molecules, again, going back to that slide at the beginning, some of those little precursor molecules can then either be converted and used to make fat or make a ketone, which then can be used to make an amino acid. So there's a back and forth. Some of those pieces can then be put in random spots in our citric acid cycle in order to help create ATP. So we do have this exchange between the two. Okay, so 3 steps to take pyruvate to get to our citric acid cycle. So we're going to remove off 1 more carbon. We end up with a 2-carbon molecule when it binds to so creating an acetyl group. When it binds to CoA, then it becomes acetyl-CoA, which is going to move into our citric acid cycle. So when that acetyl-CoA, our 2-carbon molecule, binds to our oxaloacetic acid, which is the C4 one, you're always going to end up with that last 4 carbon in this whole process. You just kind of keep going around and around. You still end up with that last 4 every single time. So you just really need, kind of get some, snatch it off with some 2 carbons and just stick it onto that 4-carbon molecule to make citric acid so I can start the cycle. So that's what we're doing when we break apart lipids in order to extract out energy. We're just stealing 2 carbons. We don't need the rest of it. We just need those 2 carbons. Okay, so we can make citric acid and start our cycle. So that's our first molecule of our citric acid cycle. So there's a bunch of stuff, blah, blah, blah. I don't care that you know this because you got other stuff to really learn about this chapter, so I'm going to keep blah, blah, blah. Who cares? Eventually, in this whole process, as you're removing off hydrogens, that's what this whole process is doing. Removing off the hydrogens, you're going to be generating some carbon dioxide that you breathe out. And then you generate that oxaloacetic acid and start this whole process all over again. So at the end of our citric acid cycle, again, I don't care what you're doing, all those little bits and pieces in the inside. So our 2 pyruvate molecules, we are going to add water to them, and we end up creating 6 molecules of carbon dioxide, which we exhale. So that's why we breathe out carbon dioxide. What's left are a bunch of hydrogen atoms that are stuck on our NAD plus and FAD coenzymes. We got to deal with them because our energy is still stuck in those coenzymes. Some of the energy is lost as heat. We also are able to make 2 ATP. And in our citric acid cycle, we do make what's called metabolites. So these are going to, again, you can convert it back and forth to other molecules that you may need. Okay, so our energy is still stuck in these coenzymes, so we got to do something about them. So we got to rip off those hydrogen atoms to transfer those energies to ATP, regenerate our coenzymes so they can go ahead and go back to either glycolysis or our citric acid cycle to pick up some more hydrogens. So what are we doing with this hydrogen? So we're moving on to the mitochondrial electron transport chain. So this whole chain is basically a bunch of enzymes that are going to pass along the electron part of the hydrogen atom. And as you're passing along, eventually that electron's going to get accepted by oxygen. And then for your proton part of your hydrogen atom, that's when we're going to build up a concentration gradient so we can use that concentration gradient to make ATP. Okay, so here are our coenzymes. Again, as you pass them along, you're reducing their energy. Oxygen is your final electron acceptor. I don't care about you knowing all of these little bits and pieces. It's just, they just keep getting passed along. Oxygen is the final electron acceptor, and we create water as technically a waste product that we actually can use. Okay, here's the nope, no, it's not. Okay, so this is all the information about them. I'm going to skip that. Okay, so what are we doing with these hydrogens? So we take these hydrogen atoms and we split them into their proton, into their electron. So the protons are technically your hydrogen ions, so they just kind of interchangeable. So these protons are pumped into the intermembrane space, so in between our inner and outer membranes. The electrons are passed along those enzymes in the electron transport chain to be finally accepted by oxygen. So as our enzymes are passing along the electrons, when a enzyme is passing electrons to another one, it's oxidized. When it receives electrons, it's reduced. So this is something more that you'll deal with like in chemistry. It's not really a big thing in my class. It's just, you just happen to hear about it here. Okay. So again, oxygen is the final electron acceptor, which is why we breathe in oxygen. So as we are pumping out protons into our intermembrane space, it's setting up a concentration gradient for a chemiosmotic mechanism. Basically, the hydrogen protons are going to go down their concentration gradient to power an enzyme called ATP synthase, and that's going to create our ATP molecules. So here's the cute cartoon version. So here in our colorful proteins in our membrane, those are our electron transport chain. So they're just passing along electrons and passing along electrons, which to be finally accepted by oxygen, creating water in the process. And these enzymes, they are pass along the electrons, but then they're pumping out the hydrogen protons into the intermembrane space, so between the outer and inner membrane. So all these protons, they start building up in this space. They go down their concentration gradient to power our ATP synthase enzyme. That's going to basically, we're attaching a phosphate group to a ADP molecule, so it has 2 phosphates, making it a TP molecule with 3 phosphates. Oh, yeah. Forgot that was there. Yay. Okay. So, again, this is our different coenzymes, so focusing on what their individual contribution is, whatever. Okay, so our complete breakdown going from glucose through glycolysis, through our citric acid cycle or Krebs cycle, and through the electron transport chain, we end up with 32 ATP at the end of it. And then the rest of it's lost as heat, but that's okay because we need to maintain body temperature. So here's a kind of broken down chart version for more text people of what's going on. So we've got glucose. So 1 molecule there. We go through glycolysis to end up with 2 pyruvate molecules. And depending on your oxygen level, so low oxygen, you go through anaerobic fermentation. Pyruvate is fermented to lactate to regenerate NAD+, so you can repeat glycolysis and make 2 ATP molecule. These lactate is what we got left over, so if we eventually those can get converted back to pyruvate. So when we have enough oxygen, or if we just started off with enough oxygen in the process in the first time, we can go through aerobic respiration. So think respiration, breathing, we have enough oxygen. So occurring in the mitochondriate, our pyruvate is converted to acetyl-CoA, so basically broken down to our 2-carbon molecule. That 2-carbon acetyl-CoA eventually is going to become citric acid, which is our first molecule in the citric acid cycle. The protons are pumped into the intermembrane space to power ATP synthase to basically attach phosphate groups to ADP to make ATP. The electrons that were passed along the electron transport chain are accepted by oxygen, creating our water, and then the carbon dioxide is just from our stripping off of that molecule and breaking down our glucose. So that's also how we end up breathing out carbon dioxide. In this whole process, we end up making 32 ATP, so way more efficient at energy production. Okay, almost done dealing with our carbohydrates. So, ATP, we use it up quickly as we can create it. So it's a energy transfer molecule, not a energy storage molecule. So we break apart that third and sometimes even second phosphate group, energy is released, and then we're able to repackage again, store some energy back in. So any excess glucose that we have, we store it as either glycogen or fat, and that's how we store energy in the body. ATP isn't used to store energy, it's just to transfer energy. So in order to take our glucose to store it as in glycogen, that process is called glycogenesis. So this is stimulated by insulin, so insulin telling your cells to soak up the glucose from our plasma or from the bloodstream, so that's going to reduce your blood sugar levels. And your process, you're going to put together molecules of glucose together to form the larger molecule of glycogen. So the way to remember this, breaking down big old words, so we have glycogenesis, so glycogen plus the creation of our glycogen, so the polymerization or the putting together of glucose into glycogen. So let's say it's been a while since your last meal, so in order to break down this stored energy, we use glycogenolysis, so the hydrolysis, so lysis, and we're using water to do so, breaking apart a larger molecule into the smaller ones, particularly taking glycogen, breaking it down so we can release glucose between the meals so we can stabilize our blood sugar, basically getting rid of hangry. So this is stimulated by glucagon and epinephrine. So remembering our epinephrine from our sympathetic nervous system. So get your body going. Glucose goes to those skeletal muscles so you can run away from the bear. So this happens when in between meals, the glycogen that's stored in the liver can be broken down, released into your bloodstream to increase blood sugar to maintain a stable level. So breaking down the big word, glycogenolysis, so glycogen plus lysis. It's not lysis, lysis. So lysis, we're breaking down. What we're breaking down is the glycogen. So breaking apart of glycogen into free glucose. Now, sometimes, again, if you're on like a low carb diet and your body's got to figure out, okay, what can we do now to get some carbs because this person didn't feed me? So that process is gluconeogenesis. So it's the creation of glucose from non-carbohydrate sources such as glycerol or amino acids. So liver, again, and also in the kidneys as well. So breaking down the big word, gluconeogenesis. So those three pieces. So it's the glucose, new creation. So we're creating new glucose from a non-carb sources. So completely different from just converting a molecule still in that same category family. Okay, so this next section is going to go into, So this is our pathway of going back and forth. I actually do have the other one. I'm just going to quickly go to it because we're about to switch into our lipid and protein, how we actually use those suckers. Ding, 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 ding. There we go. Okay, so this is what we were just talking about. So we have glycolysis breaking down our glucose. Our gluconeogenesis, so from making our glucose from non-carbohydrate sources. So that's our backwards way here. So we're able to take amino acids, they're broken down to ketones, using that to pyruvate and then going backwards. Or from our triglycerides, either as pGal or from ketones and then pyruvate, and then going back up to glucose. So non-carbohydrate sources. And then here our glycogenesis and glycogenolysis. So whether we are going back and forth from glucose to either store the glucose or release the glucose. See? I told you you're going to learn all this stuff.